Welcome to the Deep Dive. Today we're diving right into something pretty incredible in cancer treatment. Think microscopic robots. Really tiny, working inside the body to target cancer with, well, amazing precision? Sounds like sci-fi, maybe? But it's real science. We want to unpack what it means.
Exactly. And for anyone listening, the learner, this is a great chance to get a handle on a really complex but super promising field. We're talking about a potential major shift in fighting cancer. We'll try to make it clear, maybe spark some aha moments.
Yeah, absolutely. So our deep dive today comes from research looking at these intelligent nanorobot systems. And the really cool part, how they're designed to actually tweak the environment around the tumor microenvironment.
Right, the TME.
To make treatments work better. And get this, the research suggests these technologies aren't like decades away, maybe commercially ready around 2025, 2026.
That's the projection, yeah. So our mission here is pretty focused. We want to pull out the key stuff about these nanorobots, how they work, why they're potentially such a big deal, and, what hurdles are still left, all kind of tailored so it makes sense, gives you a good perspective.
Okay, let's jump in. The research really hammers home this major challenge, the tumor microenvironment, the TME. For anyone new to this, what is the TME exactly? And why is it such a problem for treating cancer?
Yeah, good question. So think of the TME as the tumor's whole neighborhood. It's not just the cancer cells. It's this whole mix of immune cells, blood vessels, structural stuff called the extracellular matrix, signaling molecules. It's busy. And the thing is, all these parts interact, often in ways that actually help the tumor, help it grow, spread, and fight off our treatments.
So it's not just sitting there, it's actively messing things up for treatment.
Yeah.
And this TME, it throws up real roadblocks for current therapies, right?
That's a huge part of it, yes. It actively hinders treatments. Like that extracellular matrix, the ECM, it can be really dense, almost like a physical wall stopping drugs from getting deep inside the tumor. And the blood vessels in tumors are often, frankly, a mess, leaky, chaotic, bad for delivering drugs properly.
And it's not just physical barriers, is it? Doesn't the TME also kind of manipulate our immune system?
Exactly. It can send out signals that basically tell the immune system to stand down, tells T cells, which should be attacking, to ignore the cancer. This immune suppression is a major reason why even things like chemo or targeted therapy sometimes just stop working effectively. They're fighting the whole supportive neighborhood, not just the cancer cells.
Wow, okay. That really paints a picture of the challenge. So how do these intelligent nanorobots come into play? How do they tackle this TME problem?
Right. So the core idea is engineering these nanorobots specifically to go after those TME challenges head on. They're not just tiny delivery trucks for drugs. They're more like multi-tool systems designed to reshape that environment, make it less friendly to the tumor.
Okay, much more strategic. The research mentions three main goals for them. Let's take the first one, dealing with those immune suppression signals. How do they do that?
So one strategy is to put PD-L1 inhibitors right onto the nanorobot's surface. PD-L1 is like a don't eat me signal cancer cells use. By blocking it with these inhibitors right there at the tumor, the idea is to basically unmask the cancer cells, letting the body's own T cells see them and attack again. It concentrates the immune boost right where you need it.
That's clever, using the body's own defenses, just enabling them better. Okay, second goal, normalizing tumor blood vessels. Why is that needed? How do they manage it?
Well, like you said, tumor blood vessels are usually abnormal, leaky, inefficient, bad for drug delivery. So the nanorobots carry VEGF inhibitors. VEGF is a signal tumors use to grow these bad blood vessels. By delivering inhibitors right to the tumor, they block that signal, aiming to make the vessels more, well, normal.
Yeah.
Healthier vessels mean better blood flow, better oxygen, and crucially, much better delivery of the drugs the nanorobot might also be carrying.
Okay. Better plumbing means the medicine gets in easier. Makes sense. And the third goal, getting past the physical barriers, the ECM. That sounds tough.
It is tough. That dense matrix is a real barrier. But these nanorobots can be designed to carry enzymes like hyaluronidase. These enzymes can actually break down parts of that matrix. Think of it like tiny enzymatic bulldozers clearing paths. By loosening up the ECM, drugs can penetrate better. Maybe even immune cells can get in more easily.
So they're actively remodeling the area. And you mentioned drugs. They also carry standard cancer drugs like doxorubicin.
Absolutely. Alongside the TME modifying agents, they're designed to carry a chemotherapy payload. Doxorubicin is a common example used in the research. This means you get that powerful chemo agent delivered with high concentration right at the tumor site, hopefully reducing side effects elsewhere.
And how do they make sure they hit the cancer cells specifically? I think the term was targeting ligands.
Exactly. Targeting ligands, like folate in some of this work, are molecules attached to the nanorobot surface. These ligands are chosen because they bind strongly to receptors that are much more common on cancer cells than on most healthy cells. It's like having a specific key, the ligand, that only fits the locks found on the cancer cells, helps them latch onto the right target.
Got it, like a molecular homing system.
Yeah.
And once they're attached, how does the drug get released? Is it just time-based or?
It's smarter than that. They use pH-sensitive linkers. These are chemical bonds holding the drug. They're designed to break apart specifically in the slightly more acidic environment, often found around tumors, maybe pH 6.5 or so. Healthy tissue usually has a more neutral pH, so this difference acts as a trigger, releasing the drug mainly in the tumor, not just anywhere.
Wow, that's a lot packed into something so small. Okay, how are they actually made? What's the design like?
Yeah, the research talks about a modular design, which makes sense given all the different jobs they have. You've basically got a module for the TME control stuff, the inhibitors, the enzymes, another module for holding and releasing the drug, and then a really important module for biocompatibility and safety, making sure the body tolerates them. This modular approach could also make them adaptable later, maybe swapping out drugs or TME agents for different cancers.
A flexible platform, then. And are they built using super experimental methods or things that are already out there?
That's actually a key point. The research emphasizes using existing, even commercially available technologies. Things like emulsion methods, co-precipitation, self-assembly. These are relatively established techniques in nanomanufacturing. This is important because it suggests scaling up production might be, more feasible than if it required entirely new exotic processes.
That's definitely good news for actually getting this to patients eventually. What about keeping them safe in the body, making sure they circulate long enough without causing problems?
Right, that's where biocompatible coatings come in. PEG polyethylene glycol is mentioned specifically. PEG is known for helping nanoparticles kind of hide from the immune system. It prevents proteins from sticking to them, which would normally get them cleared out fast. It essentially gives them a bit of a stealth cloak, letting them circulate longer and increasing the chance they'll reach the tumor.
A temporary invisibility cloak. Okay.
Yeah.
Now, this research also uses mathematical models. How do equations help design tiny robots?
The math. It's actually crucial. These models let researchers simulate how the nanorobots should behave in the body. It's incredibly complex in there. So models help predict things like release rates, how long they'll last, without doing endless, slow lab experiments for every tiny change. It helps optimize the design much faster.
Okay, there were three main equations mentioned. The first one, KEMA.
Mm-hmm.
What's that modeling?
So CAT basically models the release rate of those TME modifying agents, the VEGF inhibitors, the ECM enzymes. It helps figure out, okay, how much do we need to load? How fast should it come out to best prepare the tumor environment before the main drug hits? It's about getting the timing and dosage right for that environmental prep work.
Right, coordinating the attack. What about the second one, DDT? That sounds like the drug itself.
Exactly. DET describes the drug release rate, specifically how it responds to that pH change in the tumor. It factors in how much drug is there, how sensitive the pH linkers are. It mathematically captures that smart release trigger. This lets them fine-tune it, make sure the drug mainly gets released at the acidic tumor site, not elsewhere.
Okay, modeling the trigger. And the third one, T12 PEG. That sounds like the stealth cloak again.
You got it. T12 PEG relates to the half-life, how long the nanorobots last in the bloodstream, specifically how the PEG coating extends that time. Longer circulation time means more chance to reach the tumor. This equation helps predict and optimize how effective that PEG coating will be.
Fascinating. Using math as a virtual test lab. Okay, beyond the design and models, what's the evidence they actually work? What kind of tests did they run?
They did the rigorous testing you'd expect, started with cells in dishes in vitro, and then moved to animal models in vivo. It's a standard but essential progression.
Let's start with the in vitro stuff. What did they test on the cells?
They used various cancer cell lines like MDA, MB231, A549 grown in the lab. There, they could measure things very precisely. Did the nanorobots kill cancer cells? How well? Did they help the drug get into the cells? And importantly, did they actually change those TME factors like VEGF or PD-L1 levels at the cellular level? They'd compare the nanorobot system directly against just giving the drug alone to see the added benefit.
A direct comparison, okay. And then moving into a living system, the in vivo studies, what did those involve?
Those used mice with tumors, basically animal models of cancer. In the mice, they looked at, did the nanorobots slow down tumor growth? Did the mice live longer? Did they see those desired changes in the tumor microenvironment within the living animal? And critically, were there any harmful side effects?
And how could they see what was happening inside the mice?
They used imaging techniques, like bioluminescence imaging, to actually track where the nanorobots went in the mouse body, confirm they reaped the tumor. Afterwards, they'd also analyze tissues and fluids, look closely at the tumor to see how much drug got there, check how the immune system reacted, get the whole picture.
That sounds incredibly thorough, moving from the lab dish to a whole organism. So with all this promising data, what about getting it into actual clinics? That 2025-2026 timeline seems soon.
It does sound soon, but the research suggests it's grounded in practicality. Remember, they're aiming to use existing manufacturing methods. That's a huge plus for scaling up.
Right, not starting from scratch.
Exactly. And the materials themselves peg many inhibitors. The drugs, they often have known safety profiles already, either used in clinics now or well studied. That could potentially streamline the regulatory path compared to entirely new, unknown materials.
Okay, that makes the timeline seem more achievable. Looking further out, what's next? What's the future potential here?
Well, the big dream is personalized medicine. Imagine you analyze a patient's specific tumor, its unique TME characteristics, and then you design nanorobots tailored just for them, the right targeting ligands, the optimal mix of drugs and TME agents. That level of customization could be truly game-changing for effectiveness and reducing side effects.
Wow, truly personalized nanomedicine. Are people working on refining them even more right now?
Oh, absolutely. Constant work. Making targeting even more specific, developing smarter, maybe multi-stage drug release mechanisms, improving safety profiles even further, and always optimizing manufacturing to make it cheaper and more scalable.
And the research mentioned technology transfer. What's that about?
That's basically getting the discoveries from the university lab bench into the hands of companies. Pharmaceutical companies, medical device companies, they have the resources and expertise to run the big, expensive clinical trials needed for approval and to actually manufacture and distribute the treatment if it proves successful. It's a crucial step from research to reality.
This whole area just feels vibrant. So much potential. Let's maybe recap the big takeaways.
Sure. So these intelligent nanorobots are a really novel approach. Key thing is they don't just deliver drugs. They actively work on the tumor microenvironment, fighting immune suppression, fixing blood vessels, breaking down barriers, all while delivering drugs precisely. This could potentially overcome major hurdles like drug resistance and side effects that limit current treatments.
And hopefully for everyone listening, you now have a clearer picture of this incredible research, this potential revolution in cancer therapy that might not be too far away.
Yeah, it's a fascinating convergence of nanotechnology, biology, medicine. Really exciting stuff happening.
So here's a final thought to leave you with. We've talked about the complex engineering and deep biological knowledge needed for these cancer nanorobots. Thinking about that, what other really tough biological challenges, maybe things like Alzheimer's or maybe stubborn infections, could we potentially tackle using similar, tiny, smart, interdisciplinary approaches in the future? Something to mull over. Thanks for joining us for this deep dive.